go over the experiments that we've done as well, okay? Okay, so if you want to pause the video just to have a little look at the next slide, it breaks down all the key areas of enzymes that you need to know if you want to test yourself on it. Might be a good idea, okay? So there's your definition of metabolism, definition of enzyme, how enzymes and substrates bind together, and how they're specific, or how you can use the word specificity with them. Examples of enzymes and the substrates, so examples from your body, factors which affect enzyme activity, and enzyme experiments investigating temperature, pH, denaturation, and also immobilization. What the term immobilization means and advantages to immobilization, what the term optimum conditions mean, and how these differ for plants and animals. So girls, remember, metabolism is the sum of all the chemical reactions that happen in your body. They can be anabolic, which means it's the build-up of molecules. So molecules go from smaller ones to larger ones, such as DNA polymerase. That would be an example of an anabolic enzyme. And also catabolic enzymes, girls, are all the enzymes that occur through your digestive system. They're all breaking things down so they can be absorbed into your bloodstream, such as lipase, protease, carbohydratases. Any of those, okay, are all catabolic enzymes in your body. So remember, anabolic, think of anabolic steroids, mean building of molecules. Catabolic means breaking things down. A definition of an enzyme. So an enzyme is a biological protein catalyst that speeds up chemical reactions without getting used up, okay? So that has to be learnt off, all right? Biological catalyst, all right? Made of protein. Now, the thing about proteins, girls, is that they are really they're really strongly affected by temperature and pH. So that is why we look at how temperature and pH can affect the activity of enzymes, because they are protein. Okay. Now, so enzymes have a really specific 3D shape that allow them to bind to one substrate only. They're specific to their substrate. It's just like what's called a lock and key. Okay, so remember, every lock has a unique key, just like every enzyme has a unique substrate. So lipases, the enzyme that break down fats, will only bind fats. They will not bind proteins. They will not bind carbohydrates. Okay, and I know with my class we modelled this with plasticine, where we created two kind of like little jigsaw pieces that fit together perfectly. Now, if you change the shape of an enzyme, then it becomes what's called denatured. It no longer works, and the way we can change the shape of the enzyme is through pH and temperature. So examples from enzymes and substrates from your body, girls. So amylase, we know from your mouth, we know it's in your mouth. We know from the junior cert, breaks down starch into maltose. Protease breaks down proteins that's found in the stomach, such as pepsin. Okay, breaks down your proteins into amino acids. Lipase, then, girls, is it's a enzyme that breaks down fats, and it breaks them into their components, fatty acids and glycerol. Okay, so learn off these three, learn off their substrates, and learn off their products as well. Now, things that affect enzyme activity, we've already talked about this. So any extremes of pH, so very acidic pH, very basic pH, or very alkali pH, okay, and extremes in temperature as well. Now, there are some exceptions to the rule. So, for example, protease, which works in your stomach, pepsin, that is actually designed to work in a really low pH medium, because your stomach also produces acid. In fact, the acid in your stomach actually activates the pepsin and makes sure that the pepsin works. Okay, so that is designed to work. Its optimum its optimum pH is acidic environment. Okay, it's around pH 2 to 3. Okay, which is the pH of your stomach. So optimum, girls, is the conditions under which an enzyme works best at. We'll talk about that at the end. Now, our first experiment, girls. So our first experiment was, girls, our first set of 3 experiments, and were all very similar. It was where we used our measuring cylinders, where we added the enzyme catalase from celery. Okay, so essentially celery juice. And we added it to hydrogen peroxide, a buffer, and a tiny amount of fairy liquid. And we measured its activity by measuring the increase in foam, because hydrogen peroxide is split up by catalase into hydrogen, into water and oxygen gas. Okay, so that oxygen gas causes bubbles, which, when you have fairy liquid in there, causes foam to rise, and so the activity can be directly linked to the height of the foam in the measuring cylinder. So do you remember doing those experiments? So, girls, the way that this comes up often is they ask you what your enzyme was, what your source of your enzyme was, and what your substrate was. Enzyme catalase. It's not celery juice, okay? So your enzyme is catalase. All enzyme names will end in ASE, okay? Your substrate was hydrogen peroxide. Okay, your source of your enzyme was celery, and then also your products from this reaction are oxygen gas and water. Now, so your first experiment, girls, is where we change the pH. We change the pH, we use 4 or 5 different pH buffers, and we look to see what and we kept our temperature the same by using water bath, and we looked to see what foam height occurred for each different pH buffer. So we saw when we did a graph of it, girls, we got this kind of lovely bell-shaped curve, and when we did a graph of it, we saw that the peak of the curve occurred around pH 8, so the optimum pH for catalase is pH 8, okay? Our second experiment was looking at changing temperature. So it was done exactly the same way, except this time we used pH 8 for all of our samples, and we changed the temperature this time by using different water bath temperatures. Okay, so what we saw here, girls, was that we saw a lovely bell-shaped curve again, with a temperature of 40 degrees being the optimum temperature for the enzyme, okay? Actually, I think it was around 20 degrees. I don't know if this is the best graph in the world, but it doesn't really matter. As long as you remember, girls, that the peak of the curve is where the most activity is seen, and it's where it's the enzyme's optimum temperature there, okay? So, girls, our third experiment then, girls, this is where there can be a bit of confusion. This was the denaturation of the enzyme. That is where we heated one sample of enzyme to boiling, therefore we killed it, we denatured it, we changed its shape of its active site, and then we kept the other one at room temperature, and we measured the foam height for both of those, and we saw no foam for the denatured enzyme. We saw quite a lot of foam for the enzyme that wasn't boiled. Okay, so that shows that extremes of temperature will denature or break down an enzyme. This is a higher level experiment only, but it's the confusion can be, girls, is when you're asked a question about denaturation of catalase versus optimum temperature of catalase. Okay, remember, optimum temperature is where we do 4 temperatures. Denaturation is where we boil one sample, we don't boil the other. Now, this is probably the toughest enzyme experiment, girls, where you immobilize yeast with 2 chemicals, sodium alginate and calcium chloride. Okay, so we put yeast into little balls. Okay, yeast contains the enzyme sucrase, which breaks down sucrose, which is table sugar, to give glucose and fructose. Okay, we then put the little yeast balls into a separating funnel, and we poured table sugar solution in on top of the yeast balls into one sample. And our control sample, girls, is where we used free yeast that were not in the little sodium alginate balls, and we placed that in sucrose solution as well in a measuring cylinder. And after a number of minutes, girls, we opened up the tap on the measuring cylinder, and we tested the liquid that came out to see if glucose was present using glucose testing strips. And what we saw was that both samples created glucose. However, our product for our control, girls, was mixed with the yeast, because the yeast is so small that it just passes straight through the measuring cylinder tap. So the whole advantage, girls, of immobilizing enzymes is that they can be reused again and again and again, and they don't mix with the product. Now, so there's our immobilization, girls, means to trap something in one place, and the advantage of immobilization is that it can be the enzymes can be reused and that they do not mix with the product. And the optimum conditions, girls, finally. So optimum conditions, girls, are the conditions that enzymes work best at. They differ for plants and animals. So for animals, the temperature is 37 degrees Celsius, pH 7, okay, for most enzymes. Now, not all enzymes, remember, pepsin is one that has a preference for low pHs because it's found in the stomach. And then for plants, girls, temperature is obviously lower, 20 degrees Celsius, and around pH 9 for plants, so slightly more alkali. So hopefully that is useful, and you'll be able to use it to try and do a few exam questions on this. Thanks, girls.